Good evening, everyone. A heartfelt welcome to all participants joining us from across India and beyond. We, the students, are truly excited to present this webinar crafted with passion and purpose. On behalf of our organizing team and all biotechnology students at People's University, I warmly welcome you all to the 12th session of BioConnect webinar series. I am Neha Pandey and I am proud to tell you that I am student of BTech Biotechnology and I will be your MC for this program. It is truly a great honor to host this insightful event. Today's webinar focuses on timely and thought-provoking theme, nanosafety considerations in biotech research and applications. We are profoundly honored to extend a warm welcome to the distinguished dignitaries, thought leaders, and all participants of the program. A very special warm welcome to esteemed speaker, Dr. Maya Nair, Director of Biological Safety, University of North Texas Health Science Center, Texas, USA. Our Honorable Registrant Ma'am, People's University, Dr. Nirja Malik. Our mentor, Professor Dr. Subhash Bore, Head of the Center for Scientific Research and Development at People's University, and Mr. Gitesh Tripathi, Chairperson of the Organizing Committee for this program. To formally inaugurate today's session, it is my honor to invite our Honorable Registrar Ma'am, People's University, Dr. Nirja Malik, to deliver the opening remarks. Ma'am, over to you. Please proceed.
Thank you, Ms. Neha Pandey. Good evening to everyone joining us today. It gives me immense pleasure to welcome you all to the BioConnect webinar number 12, an inspiring student-led initiative under the Center for Scientific Research and Development at People's University. This for the students, by the students model is a shining example of how young minds can drive meaningful academic engagement when they are empowered with the right platform. Today's theme, nanosafety considerations in biotechnological research and applications, could not be more relevant. As nanotechnology continues to revolutionize healthcare, agriculture, diagnostics, and biomanufacturing, the responsibility to ensure safety, ethical conduct, and regulatory compliance becomes increasingly important. Discussions such as these help shape a future where innovation and responsibility go hand in hand. We are truly honored to have with us Dr. Maya Nair, an eminent biosafety professional from the University of North Texas Health Science Center, USA. Welcome, ma'am. Uh,
Thank
with over
you.
with over 25 years of experience in biomedical research and nearly 2 decades in leading comprehensive biosafety programs, Dr. Nair brings unparalleled expertise to this session. Her extensive work in nanoparticle based drug delivery systems and her deep involvement in institutional safety governance make her uniquely positioned to guide our students and participants for this seminar today. I also want to take a moment to appreciate the 6 enthusiastic BTech Biotechnology students who have organized this webinar with remarkable professionalism and dedication. Initiatives like these reflect the spirit of our university, where students are not only learners but also leaders and contributors to the broader scientific community. Well done, students. I must acknowledge the guidance of Professor Dr. Subhash Bore, head of the Center for Scientific Research and Development, whose mentorship and vision have helped nurture this platform for academic and societal engagements. So, kudos to you, Dr. Bore. I'm confident that today's session will expand our understanding of nanosafety and inspire our young scholars to adopt responsible and informed approaches in their research careers. With that, I extend my warm wishes for a productive and engaging webinar, and I formally declare this session open. So I hand it over to back to you, Ms. Neha. Thank you. Over to you.
Thank you, ma'am, for your inspiring words. Your unwavering support for student-led academic endeavors is sincerely valued and deeply appreciated. Now, I invite the chairperson of the organizing committee, Mr. Gitesh Tripathi, to say a few words. Mr. Tripathi, over to you.
Thank you, Ms. Neha. Hello, everyone. Our esteemed guest speaker, Dr. Maya Nair, our Honorable Registrar of People's University, Dr. Neerja Malik, ma'am, our mentor, Professor Dr. Subhash Bore, respected dignitaries, respected faculty members, and dear fellow students. My name is Geetesh Tripathi, and I am proud to tell you that I am a BTech Biotechnology student at the People's University, Bhopal. On behalf of the organizing committee, it is my privilege to welcome you to the 12th session of BioConnect webinar, a special student-led event organized by a dedicated team of BTech Biotechnology students. Today's webinar, titled Nanosafety Considerations in Biotechnological Research and Applications, focuses on a topic that lies at the intersection of innovation and responsibility. As nanotechnology becomes increasingly integrated into modern biotechnology, understanding its safety dimensions is essential for every researcher and professional in the field. We are deeply honored to have as our distinguished speaker Dr. Maya Nair, Director of Biological Safety at the University of North Texas Health Science Center, USA. Dr. Nair is a globally respected expert in biosafety, radiation safety, and nanoparticle-based biomedical research. Her extensive leadership experience makes her an exceptional resource for all of us here today. On behalf of the organizing committee, I extend heartfelt gratitude to our Honorable Registrar, Dr. Neerja Malik, for her constant encouragement and for gracing this event with her presence and opening remarks. A special thanks to our mentor, Professor Dr. Subhash Bore, for his guidance and support. I also wish to acknowledge and congratulate my fellow student organizers. Their teamwork, creativity, and commitment reflect the true spirit of People's University. We hope this session enriches your understanding and sparks new perspectives in nanosafety and biotechnological applications. With great pleasure, I welcome you all once again and I invite you to engage actively throughout the webinar. Let us look forward to an inspiring and insightful session ahead. Now, I will hand it over to our MC. Miss Neha, over to you. Thank you.
Thank you so much, Mr. Tripathi, for your encouraging words and welcoming remarks. Esteemed guests, fellow students, and distinguished participants, we now move to the heart of today's program. To take it further, now I invite the session to our moderator. Ms. Abir, over to you.
Thank you, Ms. Neha. Hello, everyone. A very warm welcome to all participants joining us from across India and beyond. I am Abir Khan, and I'm proud to tell you that I'm a BTech Biotechnology student and a member of this webinar's organizing committee at the People's University, Bhopal, India. It is my privilege to serve as your moderator for today's insightful session, organized under the BioConnect webinar series, an appreciable initiative of the Center for Scientific Research and Development at People's University. Today's session focuses on a topic of significant relevance in today's fast-evolving landscape. Today's talk will focus on nanosafety considerations in biotechnological research and applications. It is my privilege to introduce today's distinguished speaker, Dr. Maya Nair, a globally recognized expert in biological safety domain. Dr. Nair currently serves as the Director of Biological Safety at the University of North Texas Health Science Center in Fort Worth, Texas, where she also serves as a Biosafety and Assistant Radiation Safety Officer. In this capacity, she oversees a broad range of safety programs, including biosafety, radiation safety, DA control substances, animal safety, and risk management. Dr. Nair has been instrumental in establishing and leading the University of North Texas Health Science Center Biosafety Program and has served as the Biosafety Officer for the past 18 years. She has also held the role of reviewing officials for the institution Increase Control Program. With over 25 years of experience in biomedical research, Dr. Nair's scientific background is focused on nanoparticle-based targeted drug delivery system for cancer therapy. She has presented her research at numerous national and international conferences and has authored several peer-reviewed publications and book chapters on the application of nanomaterials in the biomedical field. In addition to her scientific contributions, Dr. Nair is passionate about education and training. She has developed and taught graduate-level safety courses for public health students and has mentored many students in the field of environmental health and safety. She is an active member of a university's institutional compliance group, contributing to policy development and review, and collaborates with key committees such as the Institution Animal Care and Use Committee and Institutional Review Board. Dr. Nair also serves as an external reviewer for institutional biosafety committees IVCs at other institutions. Dr. Nair has been an active member of the American Biological Safety Association, ABSA International. She is a member of the Southern Biosafety Association. Her professional interests includes the effective management of comprehensive safety programs and exploring the impact of emerging technologies on safety and security. Today, Dr. Nair will be sharing her insights on the topic nanosafety considerations in biotechnological research and applications. With this very brief introduction, it is my honor to invite Dr. Nair to deliver the talk on nanosafety considerations in biotechnological research and applications. Dr. Nair, over to you.
Thank you all, especially Dr. Subhash Bore, for inviting me to this program. Thank you, Dr. Neeraj Malik, Neha, Abir, and Geetesh for wonderful words. And it's a great endeavor what you have been doing. And this is an opportunity for you all to grow professionally. And once again, congratulate the entire team for such a great endeavor for developing you for properly handling all the safety, moreover, the research products and everything, because as you already know, the nanoparticle is booming in all different kinds of areas. So it's very, very important that we all need to work together, discuss together, study together, grow together to enjoy the fruit of this field at the same time, handle it very safely so that it will be great beneficial to the community, and we can enjoy all the results from the research involving nanosafety products. Let me share the screen. I am from the University of North Texas Health Science Center, which is one of the universities in the state of Texas in the United States. And currently, I am serving as the Director for Biological Safety Program for the university. So Fort Worth is known as the state of the Lone Star State, Texas, and the city of the West. Lot of cowboys and cowgirls, very famous for that, and longhorns and horses are very, very popular in our city. This is some snapshots from the famous Fort Worth Stockyard. And this is our downtown area. Our university is right in the middle of downtown. So this is actually from our university, this view, we can see the university. So currently, this is the university campus, what you see. Ours is mainly with health science programs. So there will be a medical college, which is Texas College of Osteopathic Medicine. And then there will be like a physician assistant program, nursing program, pharmacy program. So anything in the health science pipeline is, we have programs catering towards that health science community. As far as today's presentation, we will be going over different types of nanoparticles just briefly, and then evaluate the potential risk and concerns associated with the research involving nanoparticles, including occupational exposure, what are the parameters we need to look into, and concluding with the effective risk management strategies and an overview of global nanosafety perspectives. So what do you think about this question? Nanoparticle research should be oversight by which safety specialty? Can I see a hand gesture, like someone who think it is chemical safety? How about biological safety? How about radiation safety? How about material safety? Or it depends? Okay, let's try to answer that question together going through this presentation. So nanoparticles, it's a specific definition by ISO. What it says that it is approximately 1 to 100 nanometer size. In order to give you a comparison, our protein molecules and glucose and other molecules comes actually right at the lower level of the nanoparticle size. And then it will be like some viruses, bacteria, and cancer cell line. Just to give you an idea, cancer cell line, it's way higher, twice the size as of a nanoparticle. So it is defined 1 to 100 nanometers. So there are some examples given here, and you will in the upcoming slides you will see more about them. So this give you an overview about different types of nanoparticles and then their characteristic properties. Nanoparticle is defined based on the composition, then based on their properties, but mainly based on the composition. So there is a nanoparticle, okay, by definition, anything which fall in that measurement, 1 to 100 nanometers, is addressed as a nanoparticle. So there will be metal nanoparticles, there will be organic nanoparticles, there will be biological nanoparticles, there will be naturally occurring nanoparticles. So as the slides present, some of the examples of biological nanoparticles are liposome-based or solid lipid nanoparticles. And my area of research comes in the biological nanoparticles, lipoprotein-based nanoparticles. So it will have lipids, proteins, and then your compound of interest in the designing of the nanoparticles. So depending on the components of the nanoparticles, the properties of the nanoparticles change, both the physical, chemical, and functional properties of the nanoparticle changes. So here, as you can see, different kind of shapes in this second outer layer of circle, you can see different kind of shape. And then with different surface properties, we can engineer nanoparticles in various ways. Like, for example, when it comes to biological nanoparticles, the surface can be modified using targeted moieties like antibodies or your compound of interest. So that will help us for drug delivery, the enhanced drug delivery targeted towards specific type of cancers or specific diseases. Then there is tube-like nanoparticles, which can be like used for specific applications. Then there are nanoparticles which is engineered with specific targeted receptors, which can be directed towards specific disease again, especially in cancer treatment. These are all great advantages of nanoparticles. So summarizing, in general, the surface properties of nanoparticles by changing the charges around the molecules or by changing the functional group around the molecules or the hydrophobicity or hydrophilicity of the molecule, we can modify these molecules any way we want to. Same is possible in some of the nanoparticles, even the inner core and outside can be modified based on the way you want to design them. Like, for example, lipoproteins, the inner core will be very hydrophobic in nature. That way, lipoproteins are the carriers for lipids within our system. So like cholesterol or triglycerides, in our circulatory system, they cannot just float around like that, right? So we have to have a cargo which has the specific characteristic features so that it can contain hydrophobic materials like cholesterol or triglycerides, and then carry it to different cells through our circulatory system, which is basically pH around 7. So the inner core of the lipoprotein is hydrophobic in nature, but the outside is hydrophilic so that it can circulate through the bloodstream. And then physical properties also varies, like in different shapes, rod shape, circular shape, triangular shape. And then another major advantage is targeting. So depending on the surface properties, we can connect any kind of targeting moieties to these nanoparticles. So that provide us with a wide variety of advantages using nanoparticles in various biotechnology fields. So again, from another angle, if you look at it, the smaller size provide a wide surface area. Like nanotechnology is very popular among us. If you look around, there are several things we can see which is developed based on the nanotechnology, like some of our cream, what we are using for lotion, for skin care. So those are made out of nanotechnology. The amount of material used for taking care of skin, like lotion, for example, again, will be much less, and the delivery of the active compound will be very well controlled. So that is coming from the small size. So the same amount of active material which can be used will be much less for the purpose of the material when the nanoparticle are having the smaller size. Then surface chemistry we already mentioned. Then some of them, like the biological nanoparticles, the biocompatibility is excellent. Then like lipoprotein, once it is in our system, we really don't have to worry much about it because it's already mimicking lipoprotein particles, which we already have within our system naturally. And then another advantage is, like, when it comes to safety perspective in the research area, we actually wanted to minimize, while we mitigating the research, we wanted to minimize the exposure possibility while conducting research with nanoparticles. So this chitosan-based nanoparticle is a great example. This nanoparticle developed based on this chitosan can be used like a great delivery technique. Like some of them can be nasal delivery, some of them can be intravenous. So intravenous versus nurse, like nasal delivery, a great advantage from this risk assessment perspective because you are already eliminating the sharp usage in this case. So just to give you an example about like how to think of what are the ways to think of while we are designing that nanoparticle in biomedical research area. Similarly, like at the same time, if that it has to be intravenous or intravesical delivery, then we can be used it with greater care and proper mitigation processes. But if there is a way we can eliminate that one particular tool, here in this case it is a sharp, then we should eliminate that. That way the research, the safety is very well addressed by eliminating the one of the hazard involved in that process. A historical timeline to give you an idea about nanoparticle. It happens way before, like I'm just presenting you basically with the cancer nanoparticle area. So the drug delivery systems developed 1975 itself. There is an FDA approved cancer treatment product was already there. In the current era, when it comes to 2005, there is Abraxane, a nanoparticle developed to deliver paclitaxel, a very well used chemotherapeutic agents in cancer treatment. Paclitaxel is the challenging feature is the solubility of the product, and because of that issue, the side effect from the current formulations, which was present at the time, which is basically castor oil based formulation, was used to prepare Taxol. So in order to address that side effects and the solubility issue, an albumin based nanoparticle was developed in 2005, and that is what is Abraxane. Abraxane, the shelf life is becoming an issue, and then further research went on, and then later on there is a liposome based paclitaxel is developed, and currently there is research going on. Another example is a compound doxorubicin. So that's also another chemotherapeutic agent. Doxil is another drug which is developed based on the liposomal formulation preparation for the drug doxorubicin. Those are some of the examples. There is a lot of compound in the pipeline with respect to that. So when it comes to biotechnology, every field you can think around you will have examples of nano products. So consumer goods, several of them are there. Then there is in the agriculture field, the energy source, then healthcare and biomedical research. A lot of research is going on. Then construction, then electronic mechanisms. So we will go over some of them. So talking about nanotechnology, and the greatest gift we received was the COVID vaccines, which controlled the pandemic, like the COVID. So it was based on mRNA based COVID vaccines. Liposomal COVID vaccines were the one developed, and it was of great benefit for the humankind to control the COVID. It's not just vaccine alone. So design of PPEs, like, coating that with proper properties about disinfectants and disinfectant properties, and also the mask which we use, some of them. Then there is detection techniques based on nanotechnology. So another example is sometimes the slide. Okay. Another example is, the COVID testing, that kit you are getting for COVID testing, that also involved nanotechnology. So if you look very carefully, you can see that there is a coating of graphene here. And then, you put a drop from your, you take a nasal swab and then put it in a liquid. And that contains, the swab contains, if there is COVID, then the coronavirus will be there. And the source, which is put it into a film, and then that is colorimetrically developed by, see, the graphene and the antibody linked to that. And then the COVID can go bind to the antibody. And after some time, you develop that color. That's also based on nanotechnology. So there are several sensors developed during that time frame to identify the coronavirus. And then vaccines, different formulations. So those vaccines, majority of them are lipid-based nanovaccines. Some of them are like recombinant vector-based nanovaccines. And then not just for COVID alone, for other diseases also. So in this, you can see various diseases. They developed different kind of nanoparticle-based products to treat different kind of diseases. The cancer is a major one. Then there will be fungal infections and other kind of respiratory distress, all those distress syndrome, several of them. So these are a few current ones I'm just sharing with you to give you an example. So where are we going with this nanoparticle and nanomaterial opportunities? 2026, almost in 6-year time frame, the research, they projected to double the amount of the value of that research. So it's really going great, that present with a lot of challenges. So now let's see why do we need a drug delivery platform? So if normally this is just a schematic representation. Normally, if you want to treat a cancer, you will have the chemotherapeutic agents and you normally inject them. So when you inject them, what happened? Majority of them is getting into the liver or kidney and excreted it out of your system. And the cancer is not getting a lot of it. It's also getting a small amount like every other cells. That's why in chemotherapy regime, your immunity will be lost because your whole system will get affected by that chemotherapeutic agent. And then the other way is liver is the rescuer for our bodies. So liver trap a lot of the chemotherapeutic agents which is injected into our system. So in net effect, the cancer cell lines are getting either not effective concentration to react with the cancer cell lines, or a very minimum amount. That's what it is happening. But on the other hand, if you put that active ingredient for the chemotherapeutic agent in a specific nanoparticle or a carrier, which is targeted towards cancer. Majority of this is getting into the cancer, and then the cancer can be treated and targeted very well. So that's why we need a drug delivery system. So currently, there are a lot of nanoparticles-based treatment modalities are available there. I just highlighted the one based on different nanoparticles, like biological nanoparticles. Liposomal ones are the highest one. And then we have all other things. Micelles are there, emulsion, then inorganic nanoparticles are also there to treat cancer. So this is my area. So we use RHDL as the nanoparticle. So the components, phospholipids, cholesterol ester, and triglycerides and everything, it is very well characterized. This is our so-called good lipoprotein, which is the high density lipoprotein, which is actually removing cholesterol from peripheral tissues. So it actually a very healthy one. So we characterize the HDL and the amount of phospholipids, cholesterol, and triglyceride and everything present in the HDL particle. We use the same composition and remove the triglyceride with the hydrophobic compounds. Like in our case, it was paclitaxel or doxorubicin to pack it inside. So the mechanism is like the Trojan horse: trap the active ingredients and then send them into the cells. So the cells won't disguise that this is any active ingredient in there. And once it is, cells take it up using a receptor-mediated mechanism, it will help to kill the cells. So our lab was intensively working on this, and we have several patents on this with a lot of siRNA. The inside can be packed with various active ingredients. mRNA vaccines are also similar with liposomes. So the mechanism is through SRB1. So in this particular examples, we have paclitaxel as well as we have doxorubicin. We packed two different drugs for combination therapy, but we packed everything accordingly. And this entire nanoparticle, RHDL nanoparticle, will be taken up by the SRB1 receptor, so specific receptor mediated. And these cancer cells are overexpressing this SRB1 receptor because that is a cholesterol uptaking mechanism, and rapidly dividing cells need cholesterol. So this is towards our advantage. So it will be taken up through SRB1 receptor, and the cancer cells can selectively receive those chemotherapeutic agents, and that way the targeted delivery will be very effective. Am I going too fast?
Please go ahead.
Okay.
It's perfectly fine.
So that is about the design and application and all. So now safety aspect. Safety come into picture the minute we have that concept, how we do the work. So synthesis involved, RHDL is my example again, synthesis involved various processes. We can use some manufacturing systems, or we can go back to our old process like colloid dialysis, sonication, or through bulk mixing process. So, for example, if we use colloid dialysis here, we need to think about, when we do the risk mitigation, we need to think about the compounds and buffer we are using, how bulk volume we are using, and all those things. And if we add sonication to that, then there is another level of safety protection is required, because you have to use hearing aid process to protect your hearing system. And from here, if sonication also create aerosolization. So you need to make sure that you have a chamber in which you use the sonicator. And similarly, if you are using equipments which is already been designed in consideration with the risk mitigation processes, still you need to go through this process, the manufacturer recommended process, to make sure that there will not be any exposure of the components what is used for the RHDL drug preparation. In these cases, if it is just RHDL drug preparation, those are all the chemotherapeutic agents are the first one come in line for thinking about the safety, because chemotherapeutic agents are hazardous chemicals most of the time. And when it comes to siRNA, you need to think through, if that's the one you are trapping in RHDL, you need to think through the sequences, and then the strand, the positive or the negative strands. So depending on the strand and the charge, the the mRNA risk mitigation process will be addressed. So if you use like the mixture, like microdialysis or any of those processes, you need to make sure that all those things will be contained and labeled properly. The microfluid technology for RHDL preparation, aerosolization should be considered, and take precautionary action while you are using that technique. So approach to nanoparticle synthesis, there are two ways, top down or bottom up approach will be there. So depending on the process what you are using and what kind of nanoparticle, when it comes to nanoparticle, whether it is in the powder form or liquid form, you will be addressing those two specific synthesis process by considering all the process involved in producing that nanoparticles, and then risk mitigation will be addressed right there. So nanoparticle synthesis in top downs, there are several processes listed in here. And then in the biological, especially when we are using plant or microorganisms to create the nanoparticles, then you have to address the biological safety, chemical safety, as well as other safety components which is involved in the research development section for those particles. So to just give you an examples, there are like bacteriophages, which is another booming up area for wound care and all, trapping this bacteriophages in hydrogels. And so there will be like band-aids available with specific bacteriophages to attack infections based on different kinds of bacteria. So bulk production, when it comes to that, it comes under NIH guideline section 3D6 because you are growing virus. And if you are modifying anything, another level of safe containment and the risk assessment should be added to that process. Same is with animal virus. You could grow the virus, and then within the bioreactor, a bulk amount will be grown. Anytime when you scale up the process, that will be another level of risk mitigation you need to add to that. And then you collect the virus supernatant, and then the adjuvant is created, and then that will be used for vaccines. This is just a schematic representation. I'm not going into the details of that, just to give you an idea about how we address safety in nanoparticle synthesized using different routes, like this one for bacteria or any other microorganisms, and then using the this one is specifically for from plant bacteria, use them to incorporate the compound of interest into the nanoparticle system. Okay. Another one, to give you a specific example, is the mRNA lipid nanoparticle vaccine. So the mRNA, again, the charge outside this RNA is very, very important, because based on that, the lipid components are mainly phospholipid and cholesterol. They will align in the form of a micelle, that simply by changing the pH of the system, and then trap the mRNA into the liposomal vehicle. That is the concept behind producing this mRNA vaccines. But in this case, you have to be very careful about the mRNA you are selecting. The Institutional Biosafety Committee need to review what kind of sequence, what's the properties, positive strand or negative strand, and then different kind of phospholipids and ethanol use, where how is it producing? Is it isolated from a human product or something? Depending on that, the risk assessment need to be done and risk mitigation need to be addressed. So if you scale up that process, you can see that there will be different parameters which will come into each developmental stage. In the basic research stage, the safety and security aspect is different when it compared to the large scale production. So for example, in basic bench work, we will do the target validation, and then we identify the shelf life, and then this and then we also identify about the challenges and advantages of scaling up the process. Then the next level comes in preclinical animal work and validation of the results again, and then how the formulation development and scaling it up for for bulk production. So after the preclinical process, it will come to clinical. So in there, the legal aspects and the safety is going in parallel. So FDA approval is required for clinical trials. And for that, you need to generate a lot of data and bulk production again. So in clinical development, you have to have approvals and good manufacturing processes should be in place. There are a different set of requirements in good manufacturing process. And when you commercialize, you need to go through a different security and compliance requirement to make sure that your approved product will be out there for out there commercially for available for treatments. So those are the different in nutshell, those are the challenges. So even while developing and characterizing like the bench to like work area to bedside, bench to bedside, in that processes there are several different other analytical techniques we use. Like for example, the CTA potential will be measured for nanoparticles because we know that the surface charge is an important parameter. Then transmission electron microscopy, high resolution TEM, atomic force microscopy, and all will be used for the size and shape of the particles. Then there will be dynamic laser scattering technique for sizes to measure the size, X-ray diffraction to understand the properties of the nanoparticles. So while we are using all these techniques to analyze the nanoparticles, we need to think through what are the components of the nanoparticle and then do the risk assessment and risk mitigation accordingly. So risk management process. So in the risk management process, first and foremost is identify the hazard. What are they? How much is that? Who are doing the work? And what are all the possibilities of things can go wrong while we are handling that hazard? Then evaluate that hazard. Then we determine the control parameters and then implement the controls. And do we stop right there? No. We continue that process because it is process improvement. Every time when we come to the end of that circle, we will see, oh, which are the areas we can improve and go better. Okay. So normally in our hierarchy controls, if we can eliminate, we will eliminate. And if we cannot, then we substitute. If we cannot do both of that, then we add the controls, engineering controls first, then administrative controls, and then with PPE. So here in this case, specifically for nanoparticle-based products, short, long, medium exposure duration, and with respect to the free and bound materials, you can see the risk, high risk for a long-term exposure with the free unbound material. That is the highest risk involved with that. So the mitigation process will be highly scrutinized in this case. In order to just give you an example about when we talk about nanosafety, how complex it is, just if you read this one, this is basically a RHDL-based nanoparticle is developed with some radioactive material, some traces of radioactive materials for just to understand the radio imaging possibilities and application for the nanoparticle materials. In this case, so the major steps involved in the study: synthesis of the compound, labeled compound, and the liposomal RHDL preparation. Then cell uptake studies. Again, there is radioactive material there. Then you can see the biodistribution, which is involved the animals. So first one, chemical hazard, flammable solvents, inhalation. Second one, the biological hazard, the liposome and RHDL preparation. Aerosolization, there is chemical hazard involved. Then the next step involved radioactive material, radiation hazard. So PPE recommendations are there. Then cell culture involved, like biological hazard. Then cell culture with radioactive material preparation involving RHDL. So there is both, all the three of them are there: biological, radiological, chemical. Then there is radiation involved, radioactive waste. And when it comes to animals, it's totally different kind of entities which actually oversee the compliance for that. So I think I'm lost here. Okay. So summarizing all those into this picture, animals are there, there is cells involved, chemical hazard, aerosolization, biological radiation. This is our product of interest, the nanoparticle. So let's see how complex it is. If you wanted to think through, you have to think through all different kinds of hazard, then entities, ALAC oversee the animal research, CDC and NIH oversees the biohazards, and then state health department oversees the radiation safety program. So this is very complex. And if you want to do a research in an institution like this, currently Radiation Safety Committee need to review the protocol because it's radiation and radioactive material. Biosafety Committee need to review the protocol because it has the biological materials. Chemical Safety Committee need to review the protocol because of the chemicals what they are using. And then the Animal Care and Use Committee need to review the protocol. So those are all the institutional level reviews required. And then if you look at that, if that institution can get audited by ALAC, CDC, or EPA, or FDA, or NIH, or the Nuclear Regulatory Commission, because of all these hazards involved, that institution it can be audited by any of these agencies which oversee the safety and security and compliance. So the occupational exposure, the inhalation will take the nanoparticle to lungs and then it can get metastasized into other organs. How about if once it is getting in the circulatory system, it can reach anywhere in your body, and whatever the active ingredients that can damage your system, then it can absorb through your skin. So there are various ways you can get exposed to that. And while you are doing the risk mitigation, you need to take care of that particular component of the risk mitigation to address the safety of the person who is working with the nanoparticles. Like, for example, if it is a powdered nanoparticle, and then there is aerosolization possibilities, then one has to make sure that there is enough respiratory protection so that the nanoparticle exposure through inhalation can be very well addressed. Another thing is in the challenge of the nanoparticle when it comes to exposure, also when it comes to the targetability of the nanoparticles which we are designing. So in order to address the occupational health, if you are getting exposed with the nanoparticle, it is very difficult sometimes for the physician to understand the complexity of that, because once it is in the circulatory system, yes, all these outside properties are good for targetability. But at the same time, in the circulatory system, there is a lot of different proteins. So the same property will work against the acceleration of the process, because all these proteins can come and bind to the nanoparticle, and the expected properties of that nanoparticle can be totally changed. That is called protein corona. That is the challenge in the advancement of research involving nanoparticles. So nanomedical research, the main agencies in US which is involved in regulating that field is like CDC, NIOSH, then FDA, and then the National Institute of Environmental Health and Science. What are the parameters they are mainly looking into? The size, shape, and surface chemistry and composition of the nanoparticles. The method used like mainly in vitro, that is cell studies, and then in vivo, that is animal studies, and then there is the mathematical models. In order to give you an idea about how the nanoparticle research is really advancing, from 1984 to 2024, see, it's almost 4,000 publications are there out there now. But at the same time, the occupational health and toxicity involved nanoparticle research is only 17. See, there is a big discrepancy in this research field. That means we need to do more and more research in the occupational health area involving nanoparticles. And then some safety security consideration. This, I think, some of the information I already shared with you. It is regulated by FDA, because when it comes to clinical trials, and then EPA, because it involved a lot of toxic substances in the design of nanoparticles. Then the Consumer Product Safety Commission, because they address the safety concern if public is getting exposed to that. Then there is one National Institute of Technology initiative which develop like guidelines and nanomaterials in like they spend a lot of time to understand how the nanoparticle interact with the system and develop predictive models and all for the nanomaterials. Other regulatory agencies, like for example, Nair, it have done a lot of research involving nanoparticle. But majority of them are already been published. Then FDA, whenever they have research involving clinical trials involving nanoparticles, they share that information. And then nanomaterial registry is there. So these are all some other regulatory agencies. Mainly the Nanotechnology Research Center of Nair do a toxicology studies, exposure assessment, risk assessment, and then recommend engineering controls and PPE. So they identified the gap and then focus research to address those gaps, like hazard identification, like that complex site I showed you. You have to go to the hypothesis and then figure out what are the different hazards involved before you even start the experiment. Then think through that and then identify the hazards and start the risk assessment. Then the process involved, like RHDL, how it is synthesized, depending on the process involved of the synthesis and characterization, what are the different hazards that can be presented. Then the exposure, while conducting all those things, what are the exposure assessment. Then the risk characterization and the risk mitigation and management is addressed. So the major points come out from that is key risk is identified. Nanoparticles, because of the unknown properties, like for example, that protein corona, we can characterize everything and we can predict the properties. But the minute it is into our circulatory system, there is a lot of unknown things. Because of one example is that protein corona, the modification. And then if you are working with powdered nanoparticles or unbound materials, respiratory depositions will be there. Then it is, remember, the size is so small, so it can penetrate through skin. So those are all the important information one has to consider working with nanoparticles. The biggest challenge right now is there is not an established standard, globally established standard. There are entities in the US, in European area, in Asian Pacific area. There are entities which they collaborate and come up with different guidelines and all, but there is nothing globally available at this point. More work needs to be done to come to that point. Recommendation in any safety area, like you see the hierarchy triangle, after elimination is not possible, then you have to work with engineering controls, safe work practices, and PPE. So where this direction is going, because of the unknown properties of nanoparticles, more and more biomimetic nanoparticles are coming into the picture. So it's good and bad on that. So then you need to do the risk assessment very carefully. Then 3D printing and nanomedicine is another area. Then that bacteriophage and hydrogel is another area. Targeted drug delivery is also booming up. More and more biocompatible systems are coming up now. So this is in nutshell about the Nanotechnology Research Center summary about what are the different recommendations from their side. This is very similar to that hierarchy triangle. What kind of material you are using, what activity you are using. Depending on that, engineering controls are recommended, administrative controls are recommended, and then PPE is recommended. Like for example, dry powder. If you remember the risk assessment, the unbound material with more exposure is with the highest risk, projected risk. So that's this dry powder. So here there is more control need to be implemented compared to physically bound and encapsulated material. Safety controls. When it comes to safety controls, you have to design the facility accordingly. More ventilation is required to handle the powder, but it will be controlled. So here it is mRNA liposomal preparation. The facility will be like large scale production. Then PPE need to be outlined. Then regular health surveillance. Health surveillance is a challenge because there are a lot of unknown information with the nanoparticles. Then the process involved, when you scale up that processes, how difficult it is, automation, closed system, all those things need to be taken into consideration. Waste management is another big challenge for nanoparticles because it is mixed waste involved in that. And security measures are there when it comes to large scale production. You have to have access control, material protection, cyber security, all those. And then you have to have an emergency preparedness and business continuity measures in place to make sure that if something goes wrong, how do you address that? And how do you preserve your research or the production? So there is the WHO guidelines are available for nanosafety. It's like precautionary evidence informed approaches. And then nanosafety worldwide, there is OECD. They play a key role in human health and environmental safety aspect while manufacturing nanomaterials. And then there is the strategic approach to international chemicals management because nanoparticles involve a lot of hazardous chemicals. Then European Union is there, and also the National Nanotechnology Initiative from US, some examples. So how the control measures, like there is not really a warning hazard sign, EFPA, like there is no warning sign. So long ago, in 2007, they have done a competition, and these are the signs which got selected now. But you are free to develop your own sign, like these are various examples which are available there. Then transportation, mainly what is the chemical involved in that bulk production. Based on that, your ground shipment and air shipment is controlled by the regulatory agencies, the DOT and IATA look into that aspect. Then the spill response based on the properties and the nature. If it is powder, you need to make sure that wet wipe will be used because inhalation need to be prohibited. Then HEPA filtered vacuum should be used to clean that. Liquid spills, it's always cover the absorbent materials. You also need to consider biologicals, what involved in that, what is the biohazard involved in that. If it is radioactive material involved, you have to take into consider all those, like the spill response processes. Waste generated should be segregated and collected separately and processed separately because rad waste cannot be mixed with anything else in US. Biological waste can be, like for example, if you have some cell culture with nanoparticles with radioactive materials, you can deactivate all the biological materials with the disinfectants, and then you still need to keep it as like radioactive material mixed waste. So the waste processing will be a challenging area for nanoparticle involving research. Engineering controls, fume hoods and glove compartments should be used so that the exposure can be limited. Administrative controls, the SOP will be there for all the process involved, proper housekeeping, and then training, and then minimize, or the SOPs should be addressed in such a way that it minimizes exposure from various levels. And at the end, there should be emergency plans. In the event of exposure, what need to be done should be explained very well with the researchers. PPE, general PPE, gloves, lab coats, safety goggles. And in addition to that, various respiratory protection gear should be used based on what kind of powder you are working with. And the glove selection should be very well thought because some of these particles can be penetrated through some gloves. So nitrile gloves are recommended, but once in a while you need to test the gloves to make sure that there is no penetration or any damage to the gloves through the gloves. Medical screening and health surveillance. there is a lot of unknown area with respect to nanoparticles, but still make sure that while you work with specific hazards, understand the safety data sheet information and take actual precautions. But just in case if you get exposed, immediate first aid should be taken, and you share the information of what material you are working with your medical experts so that they will be able to provide proper guidance. And then you always report any exposure to the safety department as well as your team lead. Standard laboratory practices, which will be OSHA recommended, like everything will be same, no matter whether you use chemical hazard, biological hazard, or nanoparticles, because you need to have proper PPE, proper lab attire. Then PPE should be recommended based on the risk assessment and risk mitigation of the product involved, the process involved, and any scale-up involved. Then spill cleanup procedure should be there. Lab should be ventilated properly, and all the materials what used will be labeled and properly placed. For animal handling, there is specific information about dosage, how to inject the animals, what are the specific kind of PPE need to be used, and waste segregation is very, very important when it comes to animal studies. And then post-dosing process, how to label the cages, how to notify the ICUC and the animal facility. All those are very, very important. Good laboratory practices should be followed in this case. Then exposure monitoring program. There are several exposure monitoring programs are there. The recommended levels of exposure should be monitored very carefully, like exposure monitoring tools are their personal breathing zones, area sampling, and more, and then collect the data, analyze the data, and then address proper issues. So in summary, nanotechnology offers powerful tools for improving human health, and we all enjoy the benefit of it. And it's already a part of our life. But the most increasing challenge is it is an integrated field from various safety areas. Different hazards are involved and pose a complex, multifaceted safety landscape for this particular area. Addressing these challenges need to have a team effort. Everyone should collaborate and continue the research. So now the question again, what do you think? Nanoparticle research should have oversight by which safety specialty? Chemical, biological, radiological, material? It depends. So it needs to be a collaborative effort, a team effort, because it involved majority of those safety field. And these are some of the references. And now I'm ready for questions. I'm sorry, I hope I didn't go too fast.
Hello, madam.
Yes.
Am I audible to you?
Yes.
I am Dr. Basharat from Baba Ghulam Shah Badshah University from the Department of Microbiology.
Yes.
First of all, I wholeheartedly congratulate you. This was really a great lecture, ma'am, on nanoparticles.
Yes.
And you delivered it with enthusiasm. My
Thank
first
you.
question to you is that how nanoparticles can be used for early detection of diseases like cancer?
Sorry, can you repeat that? How nanoparticle can be used for
Early
cancer
detection of
treatment?
diseases like cancer.
Detection of diseases like cancer.
Yeah, ma'am.
So there will be like any scanning process involving cancer. So the scanning compounds or the markers, if you say, for example, a PET CT scan will be used to identify some cancer, right?
Okay.
So the compound used in that can be packed in nanoparticles, because remember, it is targeted, it can be targeted towards cancer. So I'm just, I think the example I presented is also like a scanning molecule. So say, for example, we want to identify how a specific cancer metastasizes, ovarian cancer. So
Okay.
ovarian cancer overexpresses folic acids, folate receptor. So we can develop, scanning is used based on specific dyes. So if that dye can be packed in a nanoparticle, which is engineered with a lot of folic acids outside, and then inject it into the system. So what happened is, wherever this ovarian cancer metastasized, this nanoparticle go bind that ovarian cancer cells.
Okay,
So
ma'am.
I'm just explaining a concept, okay? And then when you scan that particular organism, if it is a mice, you can see wherever it is glowing, those are all the places the cancer is metastasized.
Okay.
Does that answer your question?
Yeah. Actually, I have worked on a number of cancers from Jawaharlal Nehru Cancer Hospital, Bhopal. I worked there for at least 7 years. This
Yes.
was my major part of research.
Okay.
Sir, ma'am, my second question to you is, what is the difference between regular drug and nanomedicine action?
Oh, regular drug, there are a lot of challenges. First and foremost is, most of the cancer treatment drug is water insoluble. So you will not be inject that into our system because it just float around, right?
Yeah.
So it need to be encapsulated into specific drug delivery systems. And how that drug delivery system designed, majority is going into the liposomal formulation because that's easily manufactured. Like vaccine is an example. Liposomal vaccines are available for, like they are developing vaccines for influenza, like regular, every year the flu, the virus mutated. And one vaccine for flu you got this year will not be good for next year because it's a different type of virus. It's in the same category, but different type of virus. So you need to make sure that the vaccines developed will be like, you get projected from data, you analyze it and get the information, and then you design, you predict next year it will be this kind of mutations or like a spectrum of mutations, and then you created the vaccine. Similarly, cancer drug, and then mRNA will be used to entrap that because that will be more targeted. Similarly, if you just injection, like I think I have a slide in my presentation, like why do we need targeted delivery? If you inject any compound into your system, or if you consume any compound, then liver will be the first one which get affected. That's why when you are on a new medication, the physician will tell you 3 months you analyze your liver enzymes. That is the normal way. So if you target that drug specifically towards cancer using markers, antibody, siRNA, or any of those things, the damage to other systems like liver, kidney, and other will be much less from the chemotherapeutic agents.
Okay.
Did I answer your question?
Yeah, madam, it is okay. My last question is, ma'am, can all types of cancers can be treated with nanoparticles?
Okay. Do you want to answer that question to me?
Can all types of cancers?
No. I'm asking, what do you think after listening to the presentation?
Obviously, they can be treated.
Correct. So what are the challenges?
There are a number of challenges, ma'am.
Okay, what are they?
First, you have to understand the mechanism, how you can have to target the drug site.
Correct.
How you have to deliver the drug to the particular site. That is the
Yes.
main focus. One
Yes.
must to focus on that area. When the site can be targeted, then automatically the
So one major challenge at the moment in cancer research is in vitro. You can get the targeted receptors and compounds and everything and design. But when you go from in vitro to in vivo, there is a big unknown area. That is actually, one example is that protein corona. The minute you inject, what you are predicting there are challenges involved in getting that, translating that 100% that in vitro data into in vivo data. But there is improvement, and that area of research is growing very fast so that you can address the challenges. So one word answer to your question is yes, all cancers can be treated, because currently in US there is a big approach called like personalized medicine. I'm sure globally it is there. So what that is doing is every information we have is in a data bank. So say, for example, me as a person, I just wanted to see I'm prone to any kind of cancer. All I need to do is give a sample of the DNA, and they will analyze my DNA profile and then compare it with the existing library and see which are the markers which can be matched for cancers with my profile. Then, say for any breast cancer patients with that particular marker, they can immediately go select, oh, okay, this particular breast cancer can be treated with this particular targeted moiety, because an antibody against that marker will be good enough to design the nanoparticle. So there are like personalized medicine developing in all areas, not just cancer alone. Alzheimer's, other neurological disorders, they are developing those programs. Yes.
Abir, please go ahead. Abir.
Yes.
Hello.
Thank you so much, Dr. Nair, for your comprehensive and insightful presentation. Your talk has highlighted several crucial dimensions on nanosafety and the challenges shaping in biological research. If any other participants have some questions, they can proceed with them or use the raise hand option. Till that time, I will get with some questions. To begin with, could you elaborate, Dr. Nair, could you elaborate on the most significant nanosafety challenges currently faced in academic and industrial biotechnology settings?
Of course. So nanosafety at the moment, as I explained, there is it is a complex landscape. It involves various different safety areas. So the integration of nanosafety into trainings and protocols review is very, very important. That one example I showed you, that research involving nanoparticle with radioactive materials and chemical and everything, shows that all those safety committee need to be participating in that review. And collaboration of EHS with occupational health program is very, very important, because then you can understand the complexity and the occupational health program can be effectively designed for addressing such unknown areas of safety when it comes to different hazards involved in preparing that nanoparticles. And also risk relevant data. Collect all the data, exposures and near misses and everything should be informed and submitted, and then use that data to understand the complexity of the system. For industry, biotechnology industries, continue monitoring and collect more data for human factors, how it gets exposed. People who are working in this field, there are a lot of exposure devices are available, area monitoring devices are available. Continue, monitor and collect the data. And also safety by design principles, like understand all the parameters and hazards involved, and discussion, collaboration, even while they designing the facility, address all those information, collaborate with them. Don't just design a building and try to fit a nanosafety research into that building, those kind of information. And while transparent, and share the information. You know, you share the data, whether it is negative also, you should publish and share it.
Thank you for that perspective. Building on this understanding of challenges, let's explore how institutions can practically strengthen their safety culture. From your experience leading biological safety at UNTHSC, what key elements should institutions prioritize when developing or upgrading nanosafety programs within their laboratories?
So while you are developing a nanosafety program, integrate that safety program with all other safety program. Also, take precautionary actions and then adapt it to the existing program. Institutions should invest in governance and then training. So while you are doing the training, you need to make sure that you provide examples of complex scenarios in which nanoparticles are developed and research is conducted. So the everyday research practices, best practices should be followed no matter what hazards you are using. So when you are developing a nano safety program, prioritize structured, collaborative platform should be there to address the nano safety. And it aligns with all other existing safety programs. So that's why I presented that question, whether nano safety should be overseen by chemical safety. It's like, that famous story of 4 people came and touched the elephant. So whichever part they touched, when somebody asked, what is an elephant? The person who touched the tail told that it's like a broom, right? Yes. Have you heard that story? There was a story like that. 4 people were able to, I forgot the real story. Basically, elephant was there and 4 people was asked to go touch something, touch the elephant from different perspective, maybe in the dark. They couldn't see anything. They couldn't see the big picture, basically. So wherever they felt person who touched the trunk said that it is like a tube. So in real elephant is not anything like that, right? So if nano safety is overseen by chemical safety, they will look deep into chemical aspect. They don't look other things in that and bring that into the discussion. That's why it has to be integrated with other part of safety, and then come up with checklist, auditing checklist about what are the different hazards involved, and then all the safety compliance safety team members should discuss it annually to review and analyze the data collected. That's why it is important that any incident should be reported and analyze it. So a competency-based training should be very, very crucial in this case. You need to have regular trainings and nano safety trainings and everything, but competency-based and routine review of the protocols should be conducted. Communication is key in any safety training.
That's very enlightening. Now, considering the increasing integration of nanomaterials in emerging technologies, this assessment becomes even more critical. So how do you recommend researchers approach risk assessment when dealing with novel or engineered nanomaterials, especially when established toxicological data are limited?
Yeah. That is the biggest challenge. Toxicological data are different. So very, very limited. So in such cases, we need to consider every nano safety research is have the utmost risk, possessing the utmost risk. It is presenting with all different kind of risk. So we need to think through that process, identify the hazards and the process, and design proper exposure control. RHDL is an example. RHDL components we know, phospholipid, cholesterol, and then some organic solvents. But the way it is synthesizing and the compound which you are using to pack. So identify the hazard. Then each of those, what are the possibilities to get exposed? Then exposure control should be identified. And then provide training, adequate training to the team members, just to make sure that this is how the risk is involved, and there is a lot of unknown factors, but we are introducing proper administrative and exposure controls to make sure that this is addressed, and the risk assessment should be done along those lines, and then do the recommendations. So communication is key. Training and follow-up is very, very important in this case. And emphasize the need for the precautionary actions and the exposure control, and then the continuous risk assessment, ongoing risk assessment, and encourage them to ask questions and report. If they get exposed, anytime report. So, for example, like it is human nature that after some time you kind of, get away from PPE, like gloves I mentioned. Gloves should be checked periodically because of the small particle size that can be penetrated through the gloves. So reusing the gloves is a no with nanoparticles. Double glousing is recommended. So like that, you do the exposure control measures, and also constantly communicating and review the programs, analyze the data, and document
Thank
all
you.
these and share. Okay.
Thank you. A related concern often raised by students and early career researchers is practical containment. Could you shed light on the containment strategies and engineering control best suited for handling nanomaterials in wet labs and biotechnology facilities?
Nanomaterials in wet labs and biotechnology facilities, right? Engineering controls. Okay,
Yes.
okay. So the containment principles for nanotechnology is like the source before it get dispersed. You have to think through that, like in the research facility as well as for the bulk production. So the directional airflow is very, very important. It's always away from the workers. Okay. So when you design, when you have that, select the engineering controls, you need to work closely with the facility designers and make sure that you address all those in requirement for the directional airflow. And then if there is any filtered nanoparticles go through that airflow, like ducted versus not, like biosafety cabinets or fume hoods, then there should be another secondary trap to capture all those fine particles. And then test the performance of those equipments before you even start using it for the facilities. And any kind of aerosol generations, there should be measures taken, engineering control should be used, then there should be administrative controls and proper PPE. You should have a respiratory program. And then that program should be available for researchers and manufacturers. And there should be a system in place to make sure that they request the fit testing annually. And then also you document that data, they go through the fit testing and everything for the PPE part. And for containment, every once in a while, a trial run will be good to make sure that equipments are working properly. Follow all the requirements of certifications and maintenance for all the equipments adequately. So when it compared to wet labs and liquid nanomaterial, the risk is low, as we mentioned in the discussion. Chemical fume hoods are primary controls if you are not working with any biologicals. Then glove boxes, closed systems, and everything are the different engineering controls you can use towards mitigating the risk there. So whenever possible, we recommend the nanoparticles should be handled in liquid suspension. So if it start with a powder, handle and use the powder with proper engineering controls. And then you're at the earliest you can get into the liquid state or bound state. That will be the best way to handle the nanoparticles. Like I mentioned in all other previous questions, routine performance verification, certification, and all those, and then communication, train the team members how to use this effectively, and then some control strategies. Like, for example, we actually validate our fume hoods annually, and any time when it moves from one location to another, and the lab members are supposed to validate the fume hood airflow before they use that for any experiments.
Wonderful. As laboratories evolve, training becomes the backbone of safe operations. What type of training modules or competency-based approaches do you find effective in preparing researchers to safely handle nanomaterials?
Training. Training is a highly evolving field now. Like training and as well as the education. So, of course, the basic principles and information need to be taught in the uh, in the in the established process. Like in person is preferred. The first time you meet with the people and then explain, involve them in designing that experiment and explain. General safety training can be provided, whichever the institution prefers online. There are a lot of online modules are there, but I prefer a mixed type of training online to start with, but meeting with them in person is very, very important. Tiered training structure, scenario-based training structure is the best one. Like that example I showed you, show them such examples to address the complexity of nanoparticles involved. And give them simple examples so that they can understand how the risk assessment and risk mitigation is done in this particular research. So the general safety training followed by a specific competency-driven task force training should be there. And also the lab team lead should be in charge of monitoring first-time worker for a couple of times just to make sure that he's competent in the techniques involved in handling that nanoparticles. Nanosafety competencies for specifically address as well as the importance of integrating other laboratory practices into the nanosafety research is very important for a sustainable safety culture. And in our laboratories, we recommend always at least 2 people should be aware of what's going on, because some emergency situation occurs, we should have a backup plan and how to address the emergency scenarios. Good lab practices should be the lab personnel should be trained on good lab practices so that anytime when they get exposed or any incident occurs, they will report that. That's the most challenging area. People don't like to report. Without reporting, we don't get the data and we cannot modify the program. Reporting doesn't mean to be a punishable action. Rather, we should encourage them to report so that we can use the information to improve the communication and training. The scenario-based training is the best recommended one.
That is very helpful. Finally, let's touch upon future directions. Looking ahead, what major trends do you foresee in nanosafety research and policy, and how should students and young researchers prepare themselves to contribute effectively in this evolving landscape?
Okay, how the researchers. Let me see. So I believe we should take advantage of all the existing information and the data out there with respect to policy and exposure-driven risk assessment. Use that and then identify the weak areas. Like, for example, policy, you don't need to reinvent the wheel. So you review everything is available and use the existing resources and integrate the also any good principles from other areas. Like sustainability is another biggest area for all different safety or all different programs. So how to design a good program, and then how that can be a sustainable program. So greater focus should be given to task-based, life cycle-based risk assessment. Okay. And then you design that program into a sustainable program. Um, so it's very, very important that you collaborate with various safety professionals as well as researchers who is involved in that. So it should be a team effort that you collect the data, existing information, and then build the foundation from there. And then develop, like the other points what we discussed previously. What are the different components required for a good program? So based on that, the policy will be developed, right? Because the policy should support the research as well as to make sure that it should be done in the most compliant way, right? So there are policies which has already been tested in various parts of the world. So you just make use of that. So predictive, alternative, AI-assisted methods can be used, but always validate if when you try to get support from AI-assisted process. And also collect all the data from the research out there, like NIASH has done several research in safety aspects, and provide hands-on experience, not just the paper alone and the online. Provide hands-on experience, scenario-based, in-person, small workshops and things like that need to be done just to implement the policy as well as make awareness about this particular program. So that if you evolve the program like that, a good safety culture model will be developed. And always like the circle, develop, test, review, then analyze the data, go back the review again. So you will get a robust program. Okay.
Yes, sir. Hello, participants. Do you have any questions? If yes, please unmute yourself and ask questions.
Yes, ma'am. Uh, am I mutable? Am I audible?
Yes.
Actually, ma'am, actually, I am Dr. Hussain, assistant
Yes.
professor of this department. Ma'am, actually, I am working on a cellulosis. Please, ma'am, we have a great challenge for the production of cellulosis, actually from the fungus. This is a great challenge, ma'am, for us. Ma'am, can we use the nanoparticles for the higher production of cellulosis from the fungus? So that nanoparticles can trigger the cellulose producing genes.
I really don't know because that's not my area. From the application side, if you wanted, what is the process you are using to produce the cellulase?
Actually, I am producing the cellulases from the fungi, but the problem is that the production is less, and these cellulases are less explored. As we know that the enzymes of the target basis, I want to produce the much cellulases with the help of these nanoparticles so that they can trigger the cellulose producing genes.
Have you done any literature search? People tried it in that way?
Yes, ma'am. I have seen some one of my literature, not much. One of the paper there was, but ma'am, I thought that you can give me the best suggestion about that, so that I can use the nanoparticles for the production in future.
Would you mind sending me an email? I will read some of those information because that's not my
Sure, ma'am.
field. I
Sure,
don't know
ma'am.
how you put it. It
Sure.
should send me an email. I should be able to do some research, or I'm not a plant researcher, so I'm not familiar with that area, but I can ask around my colleagues and figure out and then answer the question. But
Thank you so much. Thank you so much.
concept-wise, if you are asking me, a gene delivery can be done using nanoparticle, the answer to the question is yes. So if you are trying to tackle any pathway using a specific gene in the process of that production, it can be done using nanoparticle also.
Yes, yes, yes,
That
yes.
is
Thanks.
the concept. But I wanted to know a little bit more about what specifically you are asking, and without having any knowledge in that field, I will not be able to answer the question currently. But send me an email, um, um, and then I should be able to get back to you.
Thanks, ma'am.
No problem.
Thanks.
If any one of you know that, is any of you are working in that area? Remember, it's a team effort. This is the platform you need to share your knowledge also, because I'm just talking from my angle, right? But nano safety, what we learn right now is
Exactly.
we have to
Actually,
calculate
actually,
all
ma'am.
different angles.
Yeah. Actually, ma'am, you have also asked the bacteriophages, ma'am. Actually, I have also worked on the bacteriophages.
Bacteriophage,
And
what do you want to do with the bacteriophage?
yes,
That we
yes,
can
ma'am.
do.
Yes, actually, the future treatment is the bacteriophage, ma'am. It is a future
Yes,
treatment. The antibiotics will be
especially
replaced.
for wound healing,
Yes,
especially
yes, yes,
for wound healing in
yes,
war
exactly.
zone and other things like bacteriophages trapped in hydrogels. They are
Oh,
like
yes,
band-aids trapped
exactly,
with
exactly.
medicine. Yes, that's
Thank you,
the
ma'am.
concept, but we have a long way to go because shelf life should be addressed, product should be produced, and then it has to go through all the FDA verification, all kinds of things.
Yes. Thank you.
Thank you.
All right, fellow students and esteemed participants, that concludes with a truly enriching discussion. On behalf of our team, I would like to extend my deepest gratitude to Dr. Nair for sharing her profound insights and expertise with us today. And to all our participants, thank you for your active engagement. With that, I now hand the session back to our MC. Thank you. Ms. Neha, over to you.
Thank you, Ms. Abhi, for moderating the session. As we draw this exciting session to a close, I now invite Ms. Ghazala Begum to deliver the vote of thanks on behalf of entire organizing team. Ghazala, over to you.
Thank you, Ms. Neha. Good evening, everyone. It is indeed an honor and a privilege to deliver the vote of thanks on behalf of the organizing team and the Center for Scientific Research and Development, People's University, as we draw the curtain on today's enriching session of the BioConnect webinar series. First and foremost, we extend our heartfelt gratitude to our distinguished guest speaker, Dr. Maya Nair, for graciously taking time out of her busy schedule to be with us today. Her insightful presentation on nanosafety considerations in biotechnological research and applications was thought provoking and inspiring. Ma'am, your insightful and impactful talk has not only broadened our perspective but also motivated us to envision opportunities in nanosafety domain. We are truly grateful for the knowledge and wisdom you have shared with us. We also extend our sincere thanks to the Honorable Registrar of People's University, Dr. Nirja Malik, for her presence and encouraging opening remarks. Ma'am, your continued support is highly appreciated. Our heartfelt appreciation goes out to all dignitaries who joined us today. Your presence has added immense value and significance to this event. We extend our heartfelt gratitude to our mentor and guide, Professor Dr. Subhash Bore. Sir, your vision, guidance, and constant support have been the driving force behind this webinar series. Your mentorship continues to inspire, empower, and motivate us. We thank our moderator, Ms. Abir Khan, for skillfully moderating the session. Our sincere thanks to the chairperson of the organizing committee, Mr. Gitesh Tripathi, for his support during the preparation for this event. I would like to take a moment to highlight the essence of BioConnect webinar for the students by the students initiative. It stands as a testament to the remarkable heights passionate learners can reach when given a platform to explore, question, and grow. Through this series, we not only gain insight from global experts, but also nurture a vibrant academic community driven by collaboration and curiosity. To our ever enthusiastic audience, thank you. Your participation, engagement, and thought provoking questions brought a energy and substance to today's session. Your presence Only your presence truly makes all the difference. And finally, we express our most profound appreciation to the management of People's University for their unwavering support and encouragement. Your belief in student led activities such as the BioConnect webinar series enables us to dream big and achieve more. With that, I now hand it over to our MC to continue with the presentation. Thank you very much. Ms. Neha, over to you.
Thank you, Gazala, for so eloquently conveying our heartfelt gratitude to our esteemed guest, our distinguished speaker, and every participant who graced this session. Dear participants, before we conclude, we kindly request you to take a brief moment, just 2 to 3 minutes, to complete the post webinar feedback form. The feedback form link is posted in the chat box. Your feedback form and suggestions are invaluable to us as they will help shape and enhance the quality of our future events. Please note that e certificates will be sent to participants who complete the feedback form. Now we'll wait for 2 3 minutes to let you complete your feedback form submission.
Thank you for the opportunity. I just wanted to see whether do you have any suggestions for me if I get to present this again? What should I do? What shouldn't I do? No suggestions? Sorry, I didn't hear.
We hope you have completed and submitted the feedback form. Please note that your e-certificates will be sent to your email ID provided in the feedback form. We sincerely thank you once again for participating in this enriching session. On behalf of organizing team, we extend our deepest gratitude to each of you for being the part of this intellectually stimulating and truly enriching session. We look forward to your participation in the forthcoming session of BioConnect webinar series. Until we meet again, stay curious, remain inspired, and keep building meaningful connections through science and learning. This is Neha Pandey signing off on behalf of organizing team at People's University, Bhopal, wishing you a pleasant evening and continued success in all your endeavors. Thank you.
Great. Thank you so much. Thank you, Subhash. Please share with me the feedbacks. Like if there is any suggestions or questions, I will be glad to get back to you.